Hi everyone. All right, this is part 2 of this week's presentation on metabolism. And the first part of the presentation, I talked about anabolic and catabolic pathways. And I talked about or covered fermentation. And the things to take away from that is that anabolic is building up of molecules and the catabolic is breaking down. Also, fermentation, it starts with glycolysis, but then ends with pyruvate then being converted into an alcohol or an acid. And I gave 2 examples of fermentation. It was the lactic acid fermentation and alcohol fermentation. So those are the end products of those each respective fermentations. Now we're going to talk a little bit about respiration. And before I get to that though, I want to continue a little bit more about catabolism. So we were talking about catabolism of sugars, okay? Or carbohydrates, which are complex sugars. And that is broken down further through glycolysis. But what happens if you've got a protein or a fat and not a sugar? Well, those can be broken down too or metabolized, but they enter at different parts or different pathways. So the proteins, which are on the left side of the slide here, in the purple box, if you follow those arrows, you're going to see that where they enter is not in glycolysis, but the proteins will then be enter at the end of glycolysis or by acetyl coenzyme A, which goes into the Krebs cycle. And you also see that proteins can also be incorporated into the Krebs cycle. So the glycolysis is not a pathway used on proteins. Also, though, fats can be metabolized. If you think that you're trying to go on a diet or you want a quick fix for energy, we usually go with the carbohydrates, we ourselves. You get a candy bar, high in sugar, and that will be processed quickly. But then again, if you're on some type of a diet or a starvation, haven't eaten for a long time, your proteins and fats then will be starting to be metabolized. And that's the same thing though too with bacteria. They will utilize fats and proteins in addition to carbs for energy. So in this case, the fats on the right hand side, if you see that, you get part of the fat molecule that will go into glycolysis, into that pathway, but the fatty acid part actually will bypass that glycolysis again and be trimmed into acetyl coenzyme A, and then from there go into the Krebs cycle. So that's just some more information I wanted to let you know about not just carbs being used, but also other macromolecules or other molecules, organic molecules, can be used as a source of food or energy. Okay, so we are going into the Krebs cycle. If you remember, that previous slide ended with the Krebs cycle. Now that we're going to focus in on that. And so we're beyond fermentation. We are now actually going into cellular respiration. Cellular respiration occurs after glycolysis, after the pyruvate is converted into acetyl coenzyme A, and then and only then is it ready to enter the Krebs cycle. And if you look at that, you can see those arrows actually go in a circle. It's a cycle. We have a carbon, a multi-carbon molecule that is then converted into a citrate. Okay, that citrate then will lose a carbon in carbon through carbon dioxide, and as it goes around this cycle, it loses carbons, oxygens, but then again we'll pick them up at the beginning of the cycle when more acetyl-CoA is introduced. Now the interesting thing about this is we do get NAD or FAD being reduced. They'll pick up hydrogens from this cycle, but also if you look at that, things are made, things are synthesized. If you look at citrate, what happens with that? Some of the citrate will then leave the cycle and become fatty acids or sterols. Again, let's go over to down to alpha ketoglutarate. That will, these are all intermediate molecules of the Krebs cycle, that will be converted into a glutamate, and then from there converted into amino acids and purines. So this stuff that's being synthesized from this Krebs cycle is going back into build things for the cells. Okay? So again, we've got the coenzymes are being reduced, which is cool. That will go on to the next part of respiration. But also I want you to be aware that in the Krebs cycle, things are also being made from some of these intermediate molecules. Okay? Now, remember I said you've got these coenzymes that have picked up these hydrogens. That NAD has picked up the hydrogen. The first thing it would like to do, they want to get rid of those hydrogens. So the first thing they did is they were lost. Those hydrogens were given up in fermentation. Now another thing that may happen if the cell is equipped with the equipment, that NADH will be able to give its hydrogens and those electrons in respiration to the electron transport chain. Okay, so now again, we've done glycolysis. I'm talking about respiration, cellular respiration. We've done glycolysis. We've gone through from there into the Krebs cycle, and then the final step of respiration, cellular respiration, is the electron transport system. And what that's made up of, I think I had talked about that in class, is a series of proteins, enzymes, and carriers that are embedded into the cell membrane of bacterial cells and even of our cells. And there are many things that go on in this electron transport system. Okay, first of all, the first part of it is the electron transport chain. So we've got, again, the embedded proteins and enzymes in the membrane. If you look at the left in this animation, it's kind of busy, but I want to break it down into smaller parts. You're going to see the NADH enter the picture. And watch what it does. It will give its hydrogen to that first enzyme. Oxidation of NADH. There go the hydrogens. At some point, the hydrogens the atoms will split, and the electrons will continue through the chain. And then at that last, right there, last enzyme, that electron is passed off to oxygen and hydrogen to make a molecule of water. So you are looking at the hydrogens being released from the NAD. You are looking at what happens to the electrons. And then also look at what happens with the protons, the hydrogen ions, once that hydrogen atom right here is split. There we go. The electrons will go one way into the pink carrier. That's the electrons. And the hydrogen ions now, they're just protons, will end up on the other side or outer side of the membrane. So now this is aerobic respiration because the electrons right there will meet up with oxygen and 2 hydrogen ions to make water. There's that oxygen, okay? That accepts the final electron. Aerobic respiration. Okay, now the important thing, another very important thing here is what happens to all those hydrogen ions there on the outside that have collected and accumulated on the outside of the cell membrane. Well, we end up building up a force on the outside, a strength of hydrogen ions on the outside. Those hydrogen ions will be dispersed into the inside of the cell membrane, where there's a lower amount of hydrogen ions. In fact, it's a little bit negative. And they do that by going through the ATP synthase, that green box at the end of the electron transport system, right there. Those hydrogen ions will come on in, and the force and the energy that those hydrogen ions release allows for the synthesis of ATP. So look at the word ATP synthase. Synthase means to synthesize. Now, where does that ATP come from? If you look at it and study this, it is coming from an ADP. That's a molecule with 2 phosphates. The energy given by those hydrogen ions coming through that ATP synthase will bond a loose phosphate to that ADP to make ATP. So we're going to see that miraculous thing happen right now. Watch these hydrogens. They give energy, and that alone P will now be bonded, that high energy bond, to ADP to form a molecule of ATP. Now, this electron transport system provides the majority of energy in a cell. Fermentation, some ATP, but aerobic respiration, a lot of ATP. And of course, our cells and bacterial cells, any living organism needs energy to maintain homeostasis. Okay? That's it in a simple nutshell. So anyhow, that is aerobic respiration there. Now, the electron transport system is also used, in addition for making ATP energy, it also can be used for other functions in the bacterial cell. That proton motive force, that force of hydrogen ions, can help make flagella move. It helps move the flagella, that idea of hydrogen ions providing a force or an energy. It also can be used to bring in organic nutrients within the cell. It can even be used, the hydrogen proton motive force, those hydrogen ions, can be used also to rid the bacterial cell of toxins that may enter the bacterial cell. So there are other uses other than making ATP in which the electron transport system is important to the bacterial cell. Okay. Now, we've been talking about organic molecules as being the donors, the electron donors for these pathways. I'm also going to talk about alternatives instead of your organic molecule, because it's all about the donors and the final electron acceptors. So that's what we concentrate on in microbiology and in the metabolic pathways here that we're talking about. So if you look at the top of this slide, what it is actually showing you, if you start out with an organic compound, in many cases it's your sugar. The organic compound can undergo fermentation, in which carbon dioxide is given away, or given off, I should say. Or if it goes cellular respiration, that organic compound can, after glycolysis, can be further broken down by the Krebs cycle and the electron transport system, in which ATP, of course, is produced. It's sounding like a broken record here, folks, I know. Sorry. But anyhow, if we look right here, the phonoelectron acceptors, we'll go way over here, can be oxygen, which makes it that aerobic respiration. But what happens if it isn't oxygen? And I talked briefly about this a little earlier last week. What happens if it's this? Okay, sulfur. Or if it's nitrate. Okay, or this. It is, or it could even be an organic molecule. That is anaerobic respiration. Anaerobic respiration. Okay? So we are seeing, if you start out with an organic compound, go through the steps and of respiration, and your phonoelectron acceptor, it can go either be aerobic or anaerobic. This is called chemoorganotrophy. Chemoorganotrophy. And if you look at that word organo, it means organic. So you start out with an organic molecule as your food source. Okay? Let's go way down here. What happens if you start out with an inorganic compound? And bacteria can do this. So if you get an inorganic compound, something that doesn't contain carbon or hydrogens, what you end up doing is it goes through the electron transport system. Also, ATP is produced, but the electron acceptors will not be oxygen. So this is saying that you start out with an inorganic compound, and then the electron acceptors will also be an inorganic compound. This is called chemolithotrophy. Rock eaters. Rock eaters is another name for chemolithotrophy, and only bacteria can do this. Take an inorganic compound and metabolize that inorganic compound as a food, and by having these anaerobic or electron acceptors, you will have as a phonoelectron acceptors. And I do take this back, there is oxygen there. You can have anaerobic or aerobic respiration starting from an inorganic compound. Okay, I do see that oxygen there. I forgot to mention that. Okay, so again, just make note that it's called chemolithotrophy when you start with an inorganic molecule, and only bacteria do that. Okay. Now, these are some reactions in which the terminal or the phonoelectron acceptors in anaerobic respiration. So we can use, if you see there, let me see, I want to see which ones. If you see there, we have ferric iron in that first one, which with the addition of electrons then will become ferrous. And it gives you examples also of the type of bacteria that do that undergo this type of anaerobic respiration. If you look at the orange boxes, we've got, wow, nitrate plus 2 electrons is nitrite. Does that sound familiar? That sounds like one of the tests we do in lab. And your Pseudomonas aeruginosa is an example of a bacterium that does nitrate reduction. And then it's showing even further on there, with further reduction with the oxidation with the addition of electrons. We've got some other examples here of these found in soil. Okay, look at this guy right here. Sulfur and 2 electrons is hydrogen sulfide. That kind of happens in the TSI slants, but this is actually an Archaean bacteria. Okay, so this guy usually grows in very, very hot thermal vents at temperatures above 100. There are no organic molecules anywhere to be found. So he basically is your rock eater down there, and the finer electron acceptors are sulfur. They to hydrogen sulfide. Finally, look at this guy right here. Okay, this is Salmonella. We did use this species in our lab, but what happens here is you can find this in the lumen of the gut, of our gut, and it's able to survive there where no other bacteria can, because what it does, it's a smart little guy, it induces the white blood cells in our gut to produce this tetrathionate right here. Okay? And you get the addition of 2 electrons, and then it will produce a substance in which the bacteria can use as their food. Okay? So this is just some examples of anaerobic respiration. Okay? Now finally, this is, I've got about 1 or 2 more slides to go. This is the nitrogen cycle revisited. And put simply, it is, you'll find nitrogen in the atmosphere. Okay, and I think I had talked a little bit about this in my talk last week. I just want to say, okay. So what happens is nitrogen makes up a high content of the atmosphere, but is not usable to us or most living organisms in that form. So we have to thank bacteria that go through 3 different stages to make the nitrogen in a more usable form for us and bacteria. Well, bacteria are what does that. So the first step is denitrification. I'm going to go back on that. The first step is, I'm going to tell you, it's nitrogen fixation. Okay, nitrogen fixation. So in this case, the nitrogen gas is converted into ammonia. So it's taking it out of the atmosphere, and these nice bacteria, a different kind of special kind of bacteria, will fix it into ammonia, NH4. Okay, from there then, we have different stages in which the bacteria may take the ammonia, and then it's converted into nitrate. Okay? From there, then converted further to nitrite. Then we get denitrification, where nitrate is reduced, and ultimately the production of nitrogen gas results. And again, both of these are aerobic. There are different types of bacteria that do this, and it actually is a combination of anaerobic and aerobic pathways, and it also is an example of chemoorganotrophy bacteria helping it go along the pathway, and also lithotrophy, which reduces CO2. So there's a lot here. Just to make it simple, I do want to say that this is where the nitrate reduction is testing for that nitrate to nitrite to see if the bacteria reduce that nitrate to nitrite. Remember, and it turns red if that's the case after you add A and B. If it hasn't turned color after you add the reagents A and B, well, maybe the bacteria did produce nitrite, but even went on further to oxidize it. And we test for that by adding zinc. So if after adding zinc it turns pink after zinc, it means no, that actually was negative for nitrate reduction. If it does not turn pink and you had no color change before in the A and B reagents, that means yes, it's positive for nitrate reduction. Okay. Anyhow, and there we are, just saying what I just said. Okay? Nitrate reduction test. All right, now, because bacteria are so special and they want to do things their way, there is an alternative carbon cycle. So in the environment, again, we do have carbon dioxide. The carbon is in the form of carbon dioxide in our atmosphere. And we have different ways in which that carbon is incorporated into the molecules that make us up. And the molecules, the organic molecules that we eat or bacteria eat or need as food to produce energy. So anyhow, there's another cycle, it's the alternative carbon cycle, in which there are archaea that do two things. Instead of the run-of-the-mill carbon cycle, they are able to make methane, which is called methanogenesis, to make methane out of hydrogen atoms, an organic molecule. They also have the ability, once that methane is made, to eat it, to break it down in anaerobic respiration, and the final electron acceptor of that is going to be sulfate and nitrite and metals, an anaerobic type of pathway. So again, these are only seen with the archaea, where their carbon is basically put in forms of methane instead of carbon dioxide or organic material. And then the methane is utilized or breakdown or utilized for energy. Okay? And then finally, we're a little word about your bacteria that undergo photosynthesis, your phototrophs. Now, their energy source, they're doing a couple things here. First of all, they initially, just like photosynthesis when you think of it, they initially will use light. And that light then will activate a center in the pigment, chlorophyll pigment, that will make or produce energy. Okay. The second part of that, phototrophs or photosynthesis, will be the production of an organic molecule. So in other words, the light in the initial stages is providing energy, in which the electrons are provided for the second part, which will then undergo photosynthesis and you'll result in your organic molecule or food. And the only thing that's a little different with bacteria that undergo photosynthesis is where the hydrogens come from. And in oxygenic, the hydrogens will come from the splitting of a water molecule. Whereas anoxygenic, the hydrogens will come from the hydrogen sulfide, which bacteria can use. Okay, so anyhow, this is where I'm going to end right now. It's getting kind of late. I do know that there's an exam coming up, and I'm going to put a review sheet out there, but I would like to remind you to also use the handout I gave, especially it's on the Krebs cycle. It's comparing and contrasting the Krebs cycle, anaerobic respiration, and aerobic respiration. And I'd like you to just kind of give it just look at it. It's going to show you, pay attention especially to what's going on in each and what is not going on in each. The fermentation does not have the Krebs cycle or the electron transport system, whereas anaerobic and aerobic respiration do. Another thing to pay attention to is also that fermentation does not need oxygen, but it is not the same thing as anaerobic respiration, okay? And if you look at that handout I gave you, you'll be able to see that just by looking at the diagrams, okay? Fermentation is not the same as the electron transport or as respiration. Fermentation is not the same as respiration because it doesn't go through Krebs cycle. It does not go through the electron transport system. And I just want to clear that up, okay? It is anaerobic, fermentation is anaerobic, but not anaerobic respiration, okay? All right, this is a lot here, and again, be on the lookout for the review for the exam 2 and some more messages I may have for you. I hope you all have a nice night, and I'll be talking to you or seeing you soon, okay? Bye.